Welcome to my YouTube channel. This is Pinky Sense Academy. You're going to revise KCSE papers. So I'm going to start with KCSE 2019 paper. Do revision questions as we look at the answers. So this is KCSE 2019 paper 2. Yes, we start with question number 1. You are told, section A, you are told that in an experiment to investigate the effect of sodium chloride on the growth rate in spinach seedlings, the seeds were treated with different concentrations of sodium chloride, and the results are as recorded in that table. So you have a table showing concentration of sodium chloride in moles per liter and percentage of spinach seeds which started to grow roots, and then the mean root length. So you are asked, from the results in the table above, explain the effect of increasing the concentration of sodium chloride. So when the concentration of sodium chloride was 0, the percentage was 99.98, root length was 17. But as you progressed adding the concentration of sodium chloride, you realize that the mean root length decreased, as well as the percentage of seeds that started to grow roots decreased. Why? Now, from the results in the table above, explain the effect of increasing concentration of sodium chloride. So, increased sodium chloride concentration will decrease the water potential, which will increase the osmotic pressure, making the water potential more negative outside the seedling, and therefore the cells up in the seedling will take in water by osmosis and dehydrate the plant, and that is why they are not able to grow well. The second questions you are asked, apart from a ruler, state 2 other equipment that would be used to determine the rate of growth of the root. If you don't have a ruler, you can use a thread, a wire, or a string, anything that can be used to measure, even a paper, a book, a pen. You can use anything. So you can use those: thread, book, pen, etc. The next question you asked, with a reason, state one part of the seedling that the student would focus on to determine the effect of sodium chloride on growth. With a reason, state one other part of the seedling that the student would focus on. So, growth mostly takes place in the shoot and also in the root. So, you can just look at a region that has active cell division. That's why the answer is the rate of growth will increase in length in the shoot. So you look at the shoot and the root. That's where growth occurs. So you can just check the region of active cell division, which is the shoot. Then here you are given another question that state the likely effect of the seedling increasing the concentration of sodium chloride to 2.20. So if the concentration of sodium chloride, the more it is increasing, the more it's affecting the seedlings and they are not able to grow, then you will find when the concentration of sodium chloride is too high, the seeds will be dehydrated. They will be dehydrated, or they will lose water, and therefore they will die or wither. The second question was, the table below shows results of blood cell counts per millimeter cubic of blood from a sample of people living at different altitudes. So here you have the red blood cells, and then white blood cells and the altitude in meters. So red blood cells and white blood cell, altitude in meters 750, They're about 4.8 and 0.45. So you can see the white blood cell, they are constant 0.45, 0.45 throughout. They have not changed in any way. But when you look at the red blood cell, the more the altitude increases in meters, the more the red blood cells increases. Now you are asked, explain the relationship between red blood cell count and the altitude. So the number of red blood cells, as you can see in this trend, they are increasing with increase in the altitude. That means, the higher the altitude, the lower the amount of oxygen or oxygen concentration. And therefore, a person living in high altitude area, they need to have a lot of red blood cells to transport oxygen. So, that is why the number of red blood cells are increasing with increase in altitude, so to increase the oxygen carrying capacity, because the higher the number of the higher the altitude, the higher the number of red blood cells to increase oxygen transportation. Then you're asked white blood cells count and altitude, the relationship between white blood cells count and altitude. So here, the role of white blood cell is to protect the body against disease-causing organism, and therefore there is no effect on the altitude. So you find that the white blood cells have to protect the body from harmful microorganisms. So their quantities of the pathogen or the amount of white blood cell is not dependent on the altitude. That's why you can see the white blood cell is constant at 0.45 counts per millimeter cubic of blood. Then you given another question. Explain why the chances of nose bleeding increases with increase in altitude in human. So you find the atmospheric pressure decreases with increase in altitude, and therefore there is imbalance between the atmospheric pressure in the outside and inside the body organ. And therefore the pressure that is generated by the heart will result in nose bleeding at high altitude, because the internal blood pressure is higher than the atmospheric pressure. Here we have number 3. You are asked to state one importance of irritability to living organism. Irritability is the ability to detect and respond to changes in the surrounding. So, living organism, irritability is important because it enables living organisms to avoid dangerous conditions or situations or harsh climatic condition. Then it also helps the living organisms to obtain a favorable condition. For example, if the temperature is too high, they are able to respond accordingly, maybe move away from the stimuli that is causing harm. If the light is too low or is too high, they are able to move because of this irritability. Then, in an experiment, students treated seedlings as illustrated below. Here you have unidirectional light, source of light, and then we have, so there is an affected tip, and then decapitated tip, that means it has been cut, and then the tip covered by an opaque cape. You are told, account the observation made in seedling 1. So seedling 1 is exposed to unidirectional light coming from one direction. What happens is this plant or this tip, the auxin will migrate to the opposite side, the side that does not have light, this darker side here, and they will cause elongation of cells. Cells will lengthen, will increase in length at this side. And that is why the shoot bends towards the source of light. So now you are told to account for the observation. For this decapitated one, okay, we asked for this one. So the positive phototropism tip phototrophism is response towards right. Now it is moving towards right, so it has positive phototrophism. That's why the shoot bends towards right. So the light causes the migration of auxin produced at the tip to the opposite side, or this darker side, as I have explained, and that makes the shoot to bend towards the source of light. Then you are asked to explain the similarity in the end result of seedling 2 and seedling 3. So this one, they remain upright because there are no auxins, and for this one the tip has been covered by a cape, and therefore auxins cannot be able to move. So even if light is brought here, it cannot be able to access the shoot tip because it is covered, and for this one the shoot is already cut. So that is why they grow upright. State the likely treatment that would make seedling 2 and 3 respond like seedling 1. So if you fit an agar block that is treated with auxin at the decapitated head of the seedling, and also removing the opaque material. Once you remove the opaque material, light will be able to pass, and therefore the plant will respond accordingly to where the light is coming from. Question number 4. You are told that in cats, the gene for fur color is sex-linked. Letter G represents the gene for ginger fur color, while letter B represents the gene for black fur color in a given cat species. These genes are codominant. Heterozygous females have ginger and black patches of fur, and their phenotype is described as tortoise shell. Now, let's first of all understand this, that in cats, the gene for fur color is sex-linked. That means it is inherited together with the sex chromosome. Letter G represents gene for ginger fur color, and letter B represents gene for black fur color. These genes are codominant. Codominant means that none of them is completely dominant over the other. That means if they are present, both of them will be expressed uniformly. Then you are told heterozygous female, that means she has ginger and this other color, which is black. And their phenotype is described as tortoise shell, with reference to information what is meant by the term codominant. So this one means that both alleles are expressed, or both of them are shown. There is no allele that is completely dominant over the other. So when present, both of them have equal chances of being expressed. So for example, they are shown equally. The ginger fur color will be expressed, and the black fur color will be expressed, and that is why we have tortoise shell. Explain why male cats with tortoise shell phenotype do not usually occur. Explain why male with tortoise shell color do not. So this gene, it's contained on the X chromosome. We have already said it is sex-linked. So it will be only transmitted by the female, and males only inherit one X chromosome from their mothers. So it will be difficult for them to have that tortoise shell. A tortoise shell female was crossed with a black male, determining the genotype and the phenotype of the offspring. So when you are crossing here, you start with the parental phenotype, tortoise for male, tortoise we've said, the person or the organism has both traits or both colors. And then for male it's black. The black will only be present in the X